Good evening, and welcome to our second lecture in our lecture series. When we were organizing our lecture series this year, we thought it might be interesting to highlight some work that goes on in some of the other Carnegie departments. For those of you who don't know, the observatory is just one of 6 departments spread across the country. In fact, we're the only one here in Southern California. But we cover all sorts of science topics, pretty much everything from earth science, biology, genetics, ecology. And so we thought we would use this opportunity to learn about something besides astronomy for once. And we decided we would start at the top, going right to our president, Matthew Scott, who's actually relatively new to the organization. He just joined us in September of last year. Prior to that, Matt spent 24 years at Stanford University, where he was a professor in 2 departments, the Department of Developmental Biology and the Department of Genetics. Most of his work at Stanford concentrated on understanding the genes that control development, and particularly understanding how damage to those genes could lead to things like birth defects and cancer. And so tonight you're going to hear more about genes and how they shape us. So help me welcome Dr. Matthew Scott.
Good evening. Thanks so much for coming out. I mean, no stars. I mean, what is this? There is dramatically, I've never spoken on a stage before where there are residual patches of blood from performances. And this is a little too biological, actually, for my taste. So it's fantastic to be here. I love this room, I love this place, and I'm really honored that you came to spend some time with me tonight. I'm a biologist. I look at beautiful animals. I love beautiful animals. I'd like to understand where they came from. And for me, as a molecular geneticist, I want to understand how they're shaped by genes. Because all those patterns, all those creatures are built by genes. And for a long time, biologists had this embarrassing situation. If we ever talked to astronomers, they'd describe their billion data points, and we'd have a graph with 7 data points. And it was embarrassing. And they'd show us things like this. This was done here at the observatories, this simulation of the start of the universe from Ben Oppenheimer and Judah Kohlmeyer, fantastic complexity and so much going on, and we were embarrassed. But in fact, we can now hold our heads up again because biology is in fact immensely rich in information content and complexity. This is wondrous, biology is wondrous, and as I've come into Carnegie and I'm starting to learn a little bit about earth science and astronomy and cosmology, I'm appreciating how much the mindset is really rather similar. This curiosity, looking for things that don't make sense as a path to discovery, trying to figure out how things work. Really exciting stuff. And you can look at your own origins. You were there. There's one of the atoms that's now in you, and there's another atom that's now in you, and you have a million stars in you, and so on. And I recently had the pleasure of visiting our huge telescopes in the Atacama Desert in Chile and seeing these incredible machines that are almost as incredible as the people who run them. What I really learned there is what fantastic pride of work our Carnegie people have in making discoveries with the machines and making the machines work incredibly well. I also, of course, what I really wanted to see was the night sky there. There's a reason countries all over the world send their astronomers to Chile to work, and this is a little time lapse that I made from one of the nights out. This is 3 hours at Las Campanas, staring up at the sky. I didn't want you to go through the evening with no astronomy, so I brought some of my own. You can see the telescopes moving. That made me quite nervous at first, but I grew adapted to it. There's Orion up there at the upper left, standing on his head. So I'm watching, as I visited this place, how the astronomers live. First of all, of course, they have to be nocturnal, and I don't, so that's an advantage to biology. But I also noticed all these parallels. So for example, when you look at pictures from Hubble, like the one at the right there, you see these gorgeous analogous structures. Here's another one. This is quite remarkable. The thing at the right is a little single-celled alga called Chlamydomonas that swims around, and you can see that it's a reflection of something out in deep space. The true thing that links biology and astronomy so dramatically is the sense of time, of deep time, of things happening slowly and sometimes quickly, sudden accelerations and decelerations of events that led to where we are now. And both biologists and astronomers are deeply interested in that history. Where did we come from? Where did planets come from? Where did life come from? And so on. So it's all about this problem of time in events. Now, for a biologist, the time is, of course, a bit shorter, although since biology is rooted in geology, we have to learn from our colleagues in these other fields to understand the starting points with our planets that led to life. So we look at time frames like this, where you can't read this mercifully, but this is 4.5 billion years of time on a linear scale here. And the parts that are of the greatest interest for sort of the formation of life and the evolution of life are highlighted in this larger colored area here, going from maybe 4 billion years, not quite, down to the present here, with a lot of very interesting events happening from 540 million years ago on, when the vast diversification of large life happened. But long before the large life was diversified in that period, the small life had evolved all the biochemistry, the enzymes, all the things that make cells work efficiently to gain from their energy sources, to shape cells, and to be able to reproduce. All of that had to happen in this much earlier period, up to, say, about 3 billion years ago. So we have creatures, that are very elaborate. They may be single-celled, but they have amazing complexity to them. All sorts of machines and structures. They can reproduce themselves. They can swim. Incredible abilities. But then, when the cells started to get together and team up, then you started getting things that were a lot more complicated, because in addition to all of the complexity within each cell, you had the complexity of assembled cells that could do a lot more things. And within a single organism, you had many more different kinds of cells joining together, the nerves and the muscles and the liver and the kidney and whatever else, doing all their parts and all the parts of the plants and so on. And of course, then these joined into ecosystems where they depended on different parts of the ecosystem for sustenance, for food, for energy, and they, of course, ate each other. So, and there are analogies in astronomy of things eating each other, lots of analogies like that. Okay, so I'm a developmental biologist at heart. That means I want to understand the processes by which you go from an egg or a seed through embryonic stages to form a creature or a plant. And that's an amazing process, right? You can see parts of it, metamorphosis of a caterpillar to a butterfly, the development of an egg into an organism. And the fundamental mystery, and one of the most amazing things to think about, is that from a little egg, a little tiny, tiny egg barely visible to the naked eye, comes, let's say, a person, and the information in that egg is sufficient that that person will look like their parents. That's amazing, right? It's absolutely amazing. And now that we know a lot more about how that happens, it's no less amazing. So embryos grow by making more cells. The cells split in half and you get 2 cells. They make the cells different because eventually you need lots of different kinds of cells, hundreds or thousands of kinds of cells, and they have to be rearranged and organized. If you melt down my right hand or melt down my left hand, it'll have all the same contents, but the pattern, you see, is a mirror image, right? The 2 hands. So that patterning, that organization of cells into a 3-dimensional pattern is crucial to forming animals that work. Now, the animal, of course, we're biased toward is the human animal. Early embryos look like this. These are pictures of human embryos going across the top here. single cell stage, 2 cells, 4 cells, and so on. You can see the diagrams that go along with that. And the colors here at the right indicate that the cells are already at these very early stages starting to take on different fates. They're forming different parts of the embryo or the extra embryonic tissues like the placenta. Okay, so that's what happens, and as people began to watch it, they were more and more mystified because most of the important things going on there were invisible to them. You didn't know about the genes, you didn't know about the proteins, you didn't know how the cells talk to each other to coordinate their activities. It was really utterly mysterious, and it seemed impossibly complex and impossible that it could be controlled, especially to give rise to a specific outcome, because the embryo of a human will give rise to a human, the embryo of a dog will give rise to a dog. And yet when they start out, they look pretty much the same. So how does that work? I'm proud to say with my new associate at Carnegie that some of the important early work looking at human embryos was done by some of the early directors of our Department of Embryology, which is a very vigorous research group to this day in Baltimore. And they established the standard description of the stages of human development that's still the reference description for everyone who looks at human embryology. And here are the Carnegie stages of human development as represented in modern work, but matching these original assignments back from a long, long time ago, early part of last century. So what has to happen as those different stages are forming is cells have to become different. So we have nerve cells, blood cells, muscle cells, fat cells, intestinal epithelial cells, bone tissue, connective tissue, all those different kinds of cells have to form, and that's part of the game. And then, as I just emphasized, the other part of the game is organizing them, which is hard to do. And those cells that have those different roles in the body of something all come from this central cell, which is the fertilized egg. So somehow as cells divide, they have to diversify and in an organized way. And they differ in all sorts of properties: their size, their contents, their rate of cell division, whether they stick to each other, whether they move around, because cells can migrate very dramatically. Again, analogies for all of these things in astronomy. If you think about it, all of these things happen. Different sizes, different contents of stars or galaxies, division and coming together of different things out in space, migration, movements of things, orbits, sticking things together to make planets and so on. Communication, well, communication implies sort of deliberate messaging, and that might start to get a little bit different in biology. A division of labor, also that implies a purpose, a usefulness of something, which might be a little different than astronomy. Shape changes of cells, production of signals emitted by cells, that certainly has lots of analogies in astronomy. Sensitivity to those signals, which varies. The cells have a metabolism, they eat stuff, they digest stuff, they make new molecules and so on. And some cells have, most cells in fact, have some sort of electrical properties, which they use in all sorts of different ways. So those are all these features of cells, and all of them come from fertilized egg, which is only a couple of hundred microns across, tiny, tiny thing, and yet in there is this incredible information content, so much information and so much elegance in how that information is packaged that for a long time it was impossible not only to understand it, but to even conceive of how information could be stored in such a tiny package. Now there is a lot in there. As many of you know, the chromosomes are linear strands of the molecule called DNA, and in a single cell that you can barely see with the naked eye, there are 2 meters of DNA rolled up into a little tangle. And even more impressively than that, the cell has to divide, which means it copies it. Now you have 4 meters of DNA. Imagine the tangles. Some of you, unlike me, have hair, so just think of what you need, to get the brush through, 4 meters of DNA. So it has to be separated because the 2 daughter cells have to end up not just with half the DNA, but equal halves of the DNA. So there are amazing processes that accomplish all that. It's machinery inside the cell that allows that perfect or near perfect copying of the DNA into 2 copies in a very tight space, and then the proper separation into the 2 daughter cells. And then the cells are dividing and they have to start to become different. So at the outset, you've got a sperm and an egg. They each bring 1 copy of the genome with them. And when they combine, now there are 2 copies of the genome in this thing called the zygote. And they form the little bundle of cells that you were just seeing, and they eventually split into placenta and the inner cell mass, which will actually make the embryo. And then there's this branching process that's quite reproducible, not at the details of every cell division, but in the general trend of cell types that will form. And I expect you to memorize this, so I'm going to leave this up here for 20 minutes. What I have boxed here in blue is the ectoderm, that's skin and neural, and all sorts of different parts that are skin and neural. And this stuff is called the mesoderm, that's ectoderm. This is called the mesoderm, that's muscle, connective tissue, and blood, always represented as red, just like the stuff on this floor. And endoderm here is lungs and gut. You know, it's basically those passages, okay? And in many different kinds of animals, most kinds of animals, there is this division, as it turns out, into these 3 groups of cell types. So you always see this kind of thing. And if you zoom in on some of this, you can find out that right here, coming from this stuff, through this stuff, through this stuff to here, you always end up with the heart forming from cells that have that ancestry. So now, with a much better description like this, you can start to say, how do these cells know to do something different from these cells? And how do all the red cells, for example, know to do something different than the cells that are following the blue pathway? Okay, what are the signals? What's the information? Who's giving the orders? And there is a lot going on in here to give those orders. It's not one central boss. It's lots and lots of crosstalk with cells saying, I'm going to do this, you do that. And what's happened over the past 30 years or so, as we've gotten a much better picture of what's in the genes, we've learned which genes are involved in telling these different cells to do what they do, which genes are involved in the communication systems between the cells, and we've learned the most amazing thing of all, which is that even though we're most interested in this process of development, that the genes that control the formation of these different tissues and the organization of them are very old. Those genes were around 500 million years ago, and pretty near the same genes are in insects and in us, in all animals. There are a lot of things that are very similar. And when you think about that, it makes perfect sense, because at what point, imagine the other scenario, which is our genes are totally different from another animal's genes. Well, when did that happen? Did a referee blow a whistle and say, okay, throw out all your old systems, we're going to start over? That's not what happens in evolution. You get gradual change, so a gene is tweaked a little bit so it makes a longer bone, or more brain, or different kind of blood or something. Slight changes, not like stop, hold, throw away everything, we're going to start over. Okay, so it makes sense that we've got this heritage that we share with all animals, and another even earlier heritage for enzymes involved in digestion, so on, that we even share with bacteria. Okay, so it reflects the history of life from the very beginning, and you can read it in the genes. And these kinds of processes, of course, have been fascinating to a lot of people for a long time. We've just had, I thought that was just a fantastic concert, by the way. I don't know if they're still here, but I thought it was just wonderful. And this, you know, I'm so honored to be the other piece of something that's bringing together art and science like this, because I think they're very similar endeavors in a lot of ways. And this man, of course, is the epitome of someone who could do that. And here's a drawing he made. He was fascinated, especially in his later years, by where people came from a developmental biology standpoint, and not just from an educational standpoint or intellectual standpoint. So I think this was early 1500s, as was this. Okay? And all of us, I think, are also, we have our own, our own reasons for being interested in this kind of thing and watching developmental processes. These two individuals are of special interest to me. Okay, so the absolute essence of biology, the fundamental thing that underlies everything we think about in biology is evolution. It's not optional. It is how everything has happened. It's how we think about everything, molecules, ecosystems, all of these things. Evolution is the most fundamental and important concept in biology. What is it? It's a simple idea. There are inherited programs of development. That's what I've just been pointing out. If you have a human egg, you have a pretty good idea of what's going to happen next. And if you have the egg of a sea urchin, you have a pretty good idea of what's going to happen next. There is a program of growth and development that forms the final creature, and that's an inherited program. It comes from mom and dad, right? And what evolution has to mean is that that inherited program changes a little bit. Can we see it changing? We sure can. If we turn the lights on, we could just look around the room. You are not identical to each other, even though you are classified as humans. There is variation due to lots of small changes in the genes in you. And those changes are passed on to a point, but then the children have changes too, and so on. So the change that happens is driven by random variation of genes. The copying of genes, as you pass them on to progeny, is imperfect. It's incredibly perfect, but it's imperfect. And then there are selective processes. Okay? One process of selection is somebody could decide they're never going to have kids, and that line stops at that point, right? Another selection, well, there are all sorts of selections. They can be imposed, as in agriculture, or hobbyists, or whatever, or they can be a sort of survival of the fittest kind of thing. Here's what Darwin loved to think about, because he was a pigeon fancier. And these are examples of pigeons. This is all one species. They will all breed with each other. And yet, as he points out here, at least a score of pigeons might be chosen, which if shown to an ornithologist and he were told that they were wild birds, would certainly, I think, be ranked by him as well-defined species. They look so different. What made them different? Pigeon fanciers made them different. They picked out weird-looking chicks and they bred them with each other until they got weirder-looking chicks, and that kept going until they had stuff like this. So you know that's happened with dogs. You know that's happened with pigeons. You know it's happened with corn and cucumbers. The things we eat now look nothing like the things that even in recent history they came from. And that's because of active people selecting things. But it doesn't have to be people. It can be a tsunami. It can be climate change. It can be whatever kind of accident happens. Because the idea that evolution is somehow perfect, that there's this survival of the fittest thing, which is a phrase I don't like very much. I think a better phrase is whatever works. Whatever works. Anything that leads to reproduction can be selected for. Okay? And sometimes, in very dramatic ways, evolution is driven by the ladies. And here are 3 examples of horribly maladaptive properties of male creatures. This guy barely lifts his head up, can't run from wolves. I mean, this is not a good fighting thing. This is embarrassing. And yet, if that's what she wants to mate with, that's what's going to be inherited in the next generation. And look at the peacock. I was staying in a place in Mexico that was one of these sort of adobe things. There's a little courtyard outside my room with 12-foot walls. And one of these guys made the mistake of flying into it, and he was trapped. He couldn't get out. You know, we had to get him out by catching him, because this is such a ridiculous bird, right? And look at this. This is these are incredibly beautiful, but this is not good for hiding from predators, right? So these are all examples of sexual selection where by whatever weird neural processes. The female, in this case, chooses the males to have exaggerated properties, and it can go the other way, of course, okay? Now, the rate of change in biology changes in the same way that in astronomy, sometimes there's stasis for a long time, nothing much happens, and then drama. Something explodes, right? A supernova, whatever. Okay? Biology, it's not usually explosions, although it can be affected by explosions. A volcano could certainly affect evolution of things nearby. You realize you could set in motion a completely new line of insects because you happen to step on some of them and not on others. That has nothing to do with fitness. It's pure luck, but the ones that survive are going to go on to breed. So in our sort of common sense feeling about the world we live in, we look at El Capitan in Yosemite and we say, man, that is stable. That is going nowhere. That is forever. And we look at these birds flitting along a beach and we think they're gone in no time. They are very transient. And we think of biology as sort of soft, transient, and geology, it's tough, you know, forever. And this is exactly backwards in most cases. Why? Because these birds reproduce, and you get more, pretty much just like these. This does not reproduce, and in fact, things do wear down. Now, we can see cases where biology evolution is rapid, but as I've emphasized, there's usually a cause of that. So here we go with dogs, okay? This is a good example of how in, say, 10,000 years, there's been this amazing diversification of dogs, and you can call them the same species, but without going into details, some of these little guys can't reach to make it the same species. And you know, you have different behaviors, of course, along with these different morphologies. So, you know, Jesus kitty, and then, you know, the original one up here, you know. So you have these things, and the behaviors are inherited too. You know that because you know lots about instincts. You know about your own instincts. You know, my mother told me when my son was young, he liked brooms. He liked sweeping stuff. He's 2 years old. Why did he like sweeping? She said, oh yeah, you did that. It was embarrassing. Okay, so why did I don't know, but something in there was wired. And you think about music, inheritance of musical abilities, which we know sometimes happens. You think about very strong, complex behaviors that are inherited. So the developmental process that gives rise to the animal is also giving rise to a structured brain that carries with it certain instincts. And you can select for or against them, like do you want a fierce dog, or do you want a stupid dog, or do you want a very affectionate dog? You know, you can select for those things, and they are genetically maintained. You know, breeds have different characters, right? Okay, although the people who have pit bulls always say how gentle they are, and I'm, you know, okay. Now, in evolution of the landscape, we have this kind of thing where in minutes, you can get dramatic transformation of a landscape. So we know geology can go very, very fast. And in biology, the fastest evolvers are the viruses. And they can change. They are actually often, they evolve to evolve. Viruses have gained benefit from changing quickly to evade the immune systems of whatever it is they're trying to affect. And so they actually get selected for their ability to change very quickly and very suddenly. And we know a lot about that because viruses are a great thing to study in the lab, and you can really explore these properties. But I want to come back to this idea again of stasis in evolution. When something works, and unfortunately cockroaches work really well, they don't change very much, and they don't change very fast. Why? Because they're under constant selection. They are, in their own inimitable way, perfect. And because they're perfect, any variation away from that will be stomped by one of you or me. And the ones that are the best are very close to a sort of standard mean. So things like that are kept static. And a gull flying or something, you know, that's perfection, and a gull that can't fly as well is less likely to reproduce as avidly. So this process of reproduction, which is has the ability to be very, very good, very close to a norm, is then reinforced by the selective process that keeps something that is good, good. Now there's some interesting examples of stasis. Here's a fossil fish called a coelacanth. And there are fossils of this fish from 400 to 700 million years ago when the Earth looked more like this, and there's plenty of water, right, in the Devonian. And the most recent fossils are from 70 million years ago. So, and they look pretty much the same as the early ones. So these fish didn't change much from 700 million years ago to 70 million years ago. But then in the early part of the 20th century, the story got even better. Because, look at that. What's that? Wait a minute. I just told you the fossils, the most recent fossils were from 70 million years ago. And then we have this. That's a coelacanth, okay? Fishermen found them. They'd been hiding, as Darwin once speculated might happen, deep in the ocean. And when they were pulled up, they looked pretty much like the fish of 700 million years ago. You know, slightly different, but still clearly coelacanths. This is one of many examples in science where not finding something, not finding the fossils from 70 million years ago to the present, is a bad thing to base your assertions on. But the relevance of this here is here's a fish that has barely changed in hundreds of millions of years, like dragonflies, like horseshoe crabs, like ginkgo trees. Things can stay the same if there's selection to keep them the same. Now this one was commented upon by no less than Ogden Nash, who said, consider now the coelacanth our only living fossil, persistent as the amaranth, which is a kind of weed, and status quo apostle. It jeers at fish unfossilized as intellectual snobs elite, old coelacanth so unrevised it doesn't know it's obsolete. So, wonderful. I wish I could write my scientific papers half as well as that. Okay, so, so we're interested in this evolutionary process, and as I've been leading up to, you'd like to understand, first of all, how normal development works, how you get from the egg to a creature. What are the control systems? How is that information encoded in genes that can be inherited? And then you'd like to understand what changes in those genes as different creatures evolve. Okay, so central idea is genes control the form of bodies. Here are a bunch of students at Stanford lined up to make a sort of chromosome. A little drama there in the courtyard of our building. Chromosomes carry information that guides the growth of embryos, and depending on what information they carry, the embryo will grow in different ways and make all these different structures. And the same, I'm underemphasizing plants because I'm an animal biologist, but the same things go on in plants. As you know, plants have immense diversity of form. All of that's genetically controlled. There are also environmental influences, but the basic starting point and the basic plant is genetically controlled. Okay, so we'd like to understand something about that. So we have to look at some of the working machinery here, and I've got to drag you into this, so be warned. This is a chromosome of the kind you've often seen. It's a big tangle. That's the 2 meters of DNA all packed up. Actually, it's part of the 2 meters because there are multiple chromosomes. And if you take that and dump it in detergent and let it spread out, you get this tangled mess of strands. Very, very tangled mess of strands. And you start to see the amount of stuff you're dealing with. And this little light goes off in your brain, as it did in the biologists who first started to see this. Wow, there could be a lot of stuff in there, and maybe there's enough information in there to do this. And you start thinking about DNA. And DNA is like a series of beads on a string. The beads, the little subunits, they're number 3 billion in the human genome. There's a lot of information in that 3 billion little subunits. And people differ from each other even siblings do, by about 1 in 1,000 of those. So you have 3 million differences from the person sitting beside you. In fact, you have 3 million differences from yourself, because you remember you get 1 copy of the genome from your mom and 1 from your dad, and they differ by 1 in 1,000. So you have 3 million variants just in yourself. And some of those variants don't mean anything. They have no consequences, and some of them are all those differences you're curious about. Muscle structure, eye color, hair quality, whatever. What kind of music you like, maybe even. Who knows? So facing this complexity, the biologists, look at this, and it's a little bit, now we have genome projects where we can get a list of all the genes. It's like a phone book, okay? And you know a little bit about them. You know the name of the gene, and you know where it is on a chromosome. So here we have the address and phone number, and that's about it. Phone books are not very information rich. And knowing from a genome project where the genes are, and you have about 20,000 of them on each of your chromosomes, well, cumulatively on all your chromosomes, but you have 2 sets. So you have 20,000 genes, and you have a list. Now, what do they do? How do you find out what they do? That's the mystery. Which of them matter for what purposes? Which of them are for muscle? Which of them are for thinking? Which of so on, okay? So changes in those genes underlie the differences between you sitting here and change over time. This is the variation I've been talking about upon which evolution can act, right? And some of the changes are not advantageous. Some of them are neutral. Doesn't matter if you have them or not, you'll still reproduce just as much. And some of them matter a lot in a certain situation. And we'd like to understand which changes lead to which outcomes. Okay. Now, these are genes. What are genes? So when the first simple organisms genes were studied, there were a few thousand genes, say in a bacterium or something. And then people looked at flies and worms, and there were about 20,000 genes. And it was obvious that we being far superior. What are we superior in? Well, not much, it turns out. But we do have swollen forebrains. So everybody started saying, well, there must be at least 100,000 genes from humans. And then as the studies went on and the counts became better, the number, the estimated true number, kept coming down. And in fact, if you extrapolated, humans had 0 genes, actually. But that's not true. It turns out they leveled out at about the same number as your average fly or something like that. And that again reflects the fact that we have this genetic heritage that we share with other animals, and the superiority we might want to point at is not something that's proportional to the number of genes. That's like saying the biggest possible bicycle is the best bicycle for you. That's not right. It's just the whole concept is wrong. It's how you use the genes and how they work together and what structures they create. And it actually doesn't take more genes to make a bigger brain. It just takes, that's one growth signal. That's not a big deal. But what about organizing it? So there are all these interesting mysteries. Now, I have to tell you a little bit of molecular biology, and one is that DNA there, a lot of that DNA is a code where each gene, in the simplest summary form, each gene codes for the formation of one kind of protein. So you have at least 20,000 kinds of protein. And proteins can be building blocks of cells, like bricks and mortar and fibers and membranes, so on. And proteins can be enzymes that carry out chemical reactions, and proteins can turn genes on and off, and proteins can be signals. Proteins are kind of the workforce of the cell, and there are tens of thousands of different kinds of proteins, each of which, the structure of each of which is encoded in the genes. So the game becomes turning on genes for the right purposes in the right places, because you don't need muscle proteins like the ones that actually move your muscles in your hair cells or skin cells. You don't need muscle proteins in your kidney. You need muscle proteins in the muscle, you need kidney proteins in the kidney, and so on. So part of the difference between these different tissues is you only turn on certain genes and other ones in those tissues stay silent. It's like checking a book out from the library or just having it there sitting on the shelf. Okay? So proteins are amazing because proteins are really complicated. And here's one where the biologists win the complexity competition. Okay, so a protein, like DNA, is a series of a chain of little subunits, little chemical subunits, little things which you've heard of because you read labels. They're amino acids. They're small chemicals, about 100 molecular weight, and a protein is a string of those joined together. Now, in our proteins, we only have 20 different amino acids. So at any position in the chain, you can have any of those 20. The 20 amino acids each are a little chemically different from each other, so depending on how you string them together, you'll end up with a different structure of protein. But look at the numbers. A protein can easily be 500 or 1,000 of these little amino acids strung together. Big proteins are actually much larger than that, many thousands. Here's a picture of a protein, by the way, one representation, where an amino acid would just be a little tiny bit of this. Okay, and they're big chunks of protein stuck together. These things have beautiful shapes, and they are machines of the cell and subunits of the cell. They're structural components of cells. They can also be motors and stuff. They're very cool. So it says here, and because of that it must be true, each one is composed of 20 different amino acids, each with a molecular weight of about 100 Daltons. A typical protein, let's say, would be 500 amino acids long in a long string. Okay? So the total molecular weight then would be 50,000 Daltons. That's sort of an aside. Okay, the number of variants possible, if you can have any amino acid at any position, you have 20 different possibilities at each position. So you have n positions, in this case 500. So the number of variants possible is n to the 20 to the nth power. So a 500 amino acid protein can have any of 20 amino acids in each position, so the total number of variations possible is 20 to the 500th power, which turns out to be about 3 times 10 to the 650th power. That's how many possible proteins of this length there could be. Now, of that essentially infinite number of possibilities, a tiny, tiny fraction actually exists in nature, and those are the ones that have turned out to be useful for something. But the cells are constantly experimenting, making variations on the proteins they make because the DNA changes randomly. You get mutations, you get a change. Let's try this one, let's try that one. So the proteins keep changing. In many cases, there are certain things about the protein, if they change at all, it's fatal to the cell. It's not tolerated. That cell will die if that protein has that kind of change. But other kind of changes are just fine. They're kind of experiments done by cells. And some of the experiments succeed. So look at this number, right? 3 times 10 to the 650 possible proteins. Estimated stars in the universe, something in this range, 10 to the 22nd, 10 to the 24th. Finally, the biologists can hold their heads up. Okay? So way up, I'd have to say. Way, way up. Okay. So what this means is that the possible structures that can be used to build cells and copy cells and control development is essentially infinite. You've got this fantastic playground due to the way these polymers, these beads on a string, are assembled together. Okay. So now we want to find out which ones matter. I'm a developmental geneticist. I want to know which of the 20,000 genes, which of those 20,000 different proteins is needed to control the growth and shape and so on of the animal. Okay, so that's the problem we set before us. We want to find the genes that control development. Those genes have to change for evolution to happen because you have inherited changes in body form and so on. Now let's go back, because I'm dragging through a lot of molecular biology here. Let's go back and look at an embryo actually developing in time lapse. This is an embryo of a fly, Drosophila, the great genetic organism used in labs since the beginning of the 1900s, 1910 or so. And what we're seeing is a time lapse here of the 6,000 cells, which are all the cells. They're all on the surface of this early embryo. It's still inside an egg, but we've unwrapped it to look at it, and we're going to watch it develop. We've got to give it the great gates of Kiev here. Each of these little balls is one cell, and the cells are moving. Some of them are moving inside in these folds, and some of them are moving around the tail. This is the head, this is the tail. Actually, the tail is now moving up behind the head. This is the stomach sort of here. And now you see body segments start to form, and then you're going to see the tail move back to where it belongs at the tail end. Meanwhile, some of the cells that have gone inside are making muscles and nerves and gut inside. They all started in the outside, they moved inside to do that. And now you have this thing that's pretty soon going to hatch and be a little sort of caterpillar-like thing that crawls away. Okay? So that's the process, and you can watch it happening, and it's amazing. And it leads to these kinds of segmented body structures that are so familiar. Now that process of going from, I didn't show you the very beginning, going from a fertilized egg to having those 6,000 cells where I started the show there, to having a fully functional larva that has a working nervous system, it has behaviors, it has sensory systems, it can eat, it has muscles, it has all those things. That entire process for a fly like that, 22 hours. It's unbelievable, okay? And then, of course, later, as though that's not enough, it goes through metamorphosis and turns into something that can fly around. So it's got 2 developmental programs packed into the same animal. And we are superior, excuse me. Now this particular image here is of a fossil from the fluorescent fossil beds in Colorado. It's 25 million years old. It's instantly recognizable. And I just want to point out the body segmentation and the fact that only certain segments have appendages. The legs come out of the thorax, there are 3 sets of legs, and the wings come out of one of the thoracic segments. So there's the division into segments, a nice clear pattern, very familiar, what happens to your picnics. And this, and here's what the fly looks like that is most often studied. And then appendages only in some places and not others. You can see the little antennae here on the head, you can see the red eyes, you can see the 6 legs and so on. Okay, so as it turns out, this is a great lab organism, and you can look for mutants that don't have the normal pattern. And if you do that really well, you get a Nobel Prize, as Christiane Nusslein-Volhard and Eric Wieschaus did for their work on this. So it's this nice, simple pattern of segmentation, which is a frequent kind of pattern in nature. You see these repeating segments, very familiar kind of thing, right? It's simple to think about how you get that. Okay, so how do you find out? Suppose you want to find genes that control development. You say, I'm going to look at that segmentation. I'm going to look for genes that control that. Well, in general, biologists, geneticists in particular, have a sort of sadistic side to them, which is not true of astronomers. And the sadistic side is they like to mess with genes and proteins and mutate them. Like you can zap them with ultraviolet light or chemical carcinogens, mutagens. So the idea is this. Here's the analogy. You want to figure out how a car works. You don't know anything about it. And what you do to find out what does what is you start to break stuff. So you let the air out of the tires, it doesn't work very well, you disconnect, you hit it with a hammer here in the windshield, now there's wind on your face, you pull off some part in here, in the guts of the thing, and now the horn doesn't work, or there's suddenly a large fire or something. So you beat away on this thing, breaking different parts and finding out what all those parts are for. That is exactly analogous to what a geneticist does with a genome. You randomly zap it with something that'll break the DNA, break the genes in some way, and you ask what doesn't happen, and then you make an inference that that particular gene you broke on that occasion was a gene you needed for muscles, because without it, the muscles don't form. And this kind of thing was done for segments. Here are the segments of a fly larva. Here's a gene that we know of now as a result of this work, and the gene is active in early embryos in these beautiful stripes. That's where the white places are where the gene is active. The dark places, that gene is not coming on. I told you genes are on in some places, not others. And when this gene isn't working, you get an animal that has half as many divisions as a normal animal. Aha! Now you don't know anything about how it works, but you've identified a gene that's very important for this part of the patterning process. And you can do this over and over and over and start to identify a lot of genes that are needed for all different parts of the pattern you're studying, or needed to make muscles, or needed to make nerves, or needed to tell if it's leap year, or whatever. And a lot of work like that, which did earn that Nobel Prize, led to this idea that as an artist does a roughing out of a scene with not so much detail initially, and then progressively fills in more detail, the genes do the same thing. The early acting genes kind of lay out roughly where things are going to be, and then once that's sort of settled the way it should be, additional genes come in and divide it into finer and finer parts of the pattern until you get something that crawls away. Now that's the segmentation process I was alluding to, but as I mentioned a moment ago, in addition to the genes that divide the animal into segments, you have to make the segments different from each other. And there's an idea from evolutionary biology that some of the ancient creatures were more like earthworms and they didn't have much difference between the segments. And then over a period of time, through evolutionary processes, you got diversification of segments. First, millipede-like things where they all have legs, kind of boring and simple, but one step ahead of the earthworm. And then the legs start to become different. Some of them turn into parts of heads, like antennae or mouth parts or whatever. And then eventually you have wings and just a few legs. You lose legs off them. So these are not just imagination. You can look in fossil record, you can look at the diversity of life now and start to piece together some of this evolutionary history. So what this suggests is that there must be genes involved in telling which segments should have a leg or a wing or whatever. So you look for genes where there are wings in the wrong places or legs in the wrong places, and you find them. So this is one of the most famous. This won a Nobel Prize for a Caltech professor named Ed Lewis, just down the street here, fabulous scientist. He had all sorts of other wonderful attributes in addition to his science, but I won't go into that now. Anyway, this flies have two wings. This fly does not have two wings. It has an extra set here, and that's because a gene that is normally active to prevent that extra set of wings from forming is damaged, and now they form. Okay, that's one kind of mutant. Here's one that I worked on when I was a postdoc. This is one where instead of the little stubby antennae that you can barely see here, you have these huge things growing out of the face of the fly. Those are legs. They're not supposed to be on the head. And this is a mutant called antennapedia. And in this case, what's happening is a gene that's normally active where you make the leg has been accidentally, due to mutation, turned on in the head. And it does what it does. It says, make a leg, and it takes over. And instead of making an antenna, you make a leg. And there are lots of variations. There are all sorts of familiar creatures that you know of with all sorts of phenotypes like this, on and on, okay? But there are also real ones, like this guy. Here's a pig with 6 legs, a piglet, okay? So these are we call these birth defects, right? And in fact, it's no surprise at all to you that developmental biologists end up studying genes that, if damaged, will lead to a variety of birth defects. And one of the nice things about working on flies is you don't get too emotionally involved with them, which is good, although it has happened. But that's a long story. I won't tell that story. Okay. But the point is that this study of insects led to discovering lots of genes that control development. And that's great. That's as far as anyone expected it to go. And then the astounding thing happened, which I already explained, which is that it turned out it was the same genes in us. And nobody expected that, even though we should have. No one said it. Usually you can go back in scientific history and some lunatic somewhere said everything. And you just find some book somewhere where somebody who was losing it or something said something that turned out to be true, even though they were saying it without any basis whatsoever. In this case, nobody even said that, of course, the program of development must be ancient and conserved, because when would you throw it out and start over? Obvious thing. Anyway, one of the most dramatic and earliest examples of this were these genes that control the differences between different parts of the body along the head to tail. These are the genes that put wings and legs in the right place and so on. And it turns out there's a whole set of these genes. That's what these different color boxes represent, the set of genes along the chromosome. They have the remarkable property that they're organized along the chromosome in the order of the body parts they affect. So you can see this one is right up at the tip of the head. This one at the right is the tip of the abdomen, and everything's laid out in between, which is really weird, and we still don't understand it, really why that is. But it's something about the history of the evolution of these genes. So the different properties of different parts of the body depend on the activities of these different genes. So this gray gene causes this gray part of the body to have certain properties. Okay? So that was insects, and then it turned out same story for us, or this is a mouse embryo, but it's the same for us. We can claim some superiority because we have multiple copies of the cluster of genes, like backups, and some of them are incomplete copies. But anyway, it's the same deal. The genes are active along the head to tail axis of the animal in the order that they are in the chromosome. What do they do in us? They don't put wings on any of our segments, even when you mutate them. So what do they do? Well, they control all sorts of things. They control the pattern of your digits, the patterns of your toes, the difference between your digits and toes, the differences between your vertebrae, the differences between different parts of the brain, differences in different parts of the skin. They form like an informational map that direct differences in structure across the body, mostly, but not always, along this head to tail kind of axis. So how do you demonstrate that? Well, in a mouse, you look at mutants. So here is a normal mouse skeleton with all the vertebrae lined up here, and there are, as there are in us, different classes of vertebrae. These lumbar vertebrae do not have ribs on them. The sacral vertebrae do not have ribs on them. The vertebrae up here, the thoracic vertebrae, do have ribs on them. If you mutate a bunch of these so-called Hox genes, these genes that give identity to different parts of the body, then you get amazing differences. So here, look at these ribs growing where there shouldn't be any ribs. Even back here in the sacral regions, there are extra ribs growing. Okay? So without going into lots of descriptions like this process of you look in a simple organism, relatively speaking, it's not really simple, that you can work on in the lab. You look for genes that control development in that. Then you find out that the human or other mammalian ones do similar kinds of things. And then you start trying to figure out how they work. So let's ask that question. How does one gene, just plain with one gene, cause an entire leg with all its complexity to form on the head where it shouldn't form? So I told you part of the answer. That gene is normally not active in the head, and when you turn it on in the head through a mutation, now it does what it normally does. But how does it do what it normally does? You would think you would have to control and coordinate the activities of dozens or hundreds or thousands of other genes to make something as complicated as a leg. Ha! Well, that's the answer. That's what it does. It controls hundreds of other genes. So these Hox proteins, when you produce them in a cell, it'll change the activities of dozens or hundreds of genes, thus changing the property of all the cells where it is. How does it do that? Let's look at a chromosome that's splayed out now. Here are the subunits of the DNA, and then if you follow it back, here's a chromosome, and we're sort of amplifying a little bit of it and spreading it out and uncurling it and uncurling it. Here's the famous double helix, and what the double helix really is a line of A, T, C's, and G's. That's the code. Okay, so there's a gene. A section of this is a gene, and you want to turn on that gene in a certain place in the animal. Well, things like these Hox proteins do that. So here we have these two Hox proteins that have landed on specific sequences of these A, C, T's, and G's. They recognize them, they land there, and by landing there, they turn on the gene in that vicinity. And since they can land at 100 different places, they can turn on 100 different genes. The docking of the protein, the controlling protein against the DNA, is perfect. It is absolutely perfect. Here's a picture. The DNA is in green and brown, that's the double helix, and a blue protein is stuck here, a pink protein is stuck here, and you can see it nestles right into the crack and exactly matches up with that piece of DNA. It will not bind to something it shouldn't bind to, okay? So there's this powerful force there. There are lots of proteins that do this kind of thing, and they control development. So here is a vertebrate eye, and here is a fly eye, and they couldn't be more different. Fly eyes are compound eyes, lots of lenses and so on, single lens, so on. But looking for mutants again, there's a Drosophila gene that has no eyes. There's a mouse gene, a Drosophila mutant that has no eyes. There is a mouse mutant that has very small eyes. And there is a human disease called aniridia where the iris is damaged. It turns out those are all the same gene. They're all the same gene. Evolved apart, 500 million years apart, but the same gene. The gene is needed for eyes in insects, eyes in us, eyes in mice, eyes in clams. Seriously. Eyes in all these animals. So it's a very old gene that was involved in forming some sort of light-sensing organ, and it's been conserved over all that time. And you can test its power to build an eye in a fly by turning the gene on in places it shouldn't be. Like it should be on in the eye, of course, but you can engineer it so it turns on in extra places, and you get things like this with a leg, and there's an eye growing out of the side of the leg. Okay, so just turning on that one gene in the leg causes an eye to develop there. This is another one of those controller proteins, like the Hox protein, that can bind to many genes and turn on, in a coordinated fashion, all the genes you need to build an eye. There are similar things for muscle. Here's a picture of Vesalius from the 1500s, and he did these fantastic illustrations of musculature. I think all modern scientific literature should use figures that show beautiful bridges and rivers in the background of a horribly dissected foreground. It's not horrible, it's actually beautiful, but it is a remarkable style. Now, muscles, of course, play a prominent role in our culture. Some of you know Jesse Ventura, the Minnesota governor, had this alter ego. This is him in his other guise, and that led to these bumper stickers that my governor can beat up your governor, but we're proud Californians and we can't stand for that sort of thing. So we responded appropriately. So it turns out that muscles, despite all their popular stuff, they too are controlled by ancient conserved genes that you can find. So, and particularly heart genes. So in insects, there are hearts that pump from one end to the other just as they do in us. The most impressive heart I've sort of come across is the blue whale's heart. Here's a diver for scale. Here's a person for scale in this model of a blue whale's heart. The heart is about the size of a Volkswagen. And that heart and the heart of an insect are both controlled by one of these controller proteins that can turn on a whole set of coordinated genes that you need to build a heart. And it doesn't really matter what the size is, you're still getting, this heart looks a lot like ours actually. So again, size is not the issue, it's the design and patterning. The gene was discovered in flies because the mutants didn't have hearts, and it was named Tin Man after, you know, one of our favorite characters from down the street here, who at least thought he didn't have a heart. Although in the case of the mutants, they really don't actually have hearts. Now, is this of any use? I haven't told you anything about all this work that's useful. And I do want to mention that it actually is. I mean, as in astronomy, part of what you're trying to do is just understand. And it's fascinating, and it's fun, and it's full of surprises. And working in the lab is incredible fun, and working in telescopes is even more fun. But the other part of it is, can you learn something that might be put to use? And of course, here we're talking about agriculture and medicine for biology. And in this case, here's this gene found in flies because they don't make a heart. And you look and you find a corresponding human gene that you need for heart development. And then you find out that there are families who inherit damage to that gene. What's wrong in those families? They have atrial septal heart defects. This is the Tin Man gene. It's also called NKX2.5. And they have defects in the divisions of the heart chambers. And it's a very common kind of heart birth defect. So you learn the genetic basis for a human heart disease from studying the growth of hearts in a fly. Okay? All right. It is true that I will eventually finish this talk. I know some of you are skeptical about that, but I have one more thing to tell you, which is that if it was as simple as just turning on or off batteries of genes within a cell, you'd have no coordination between cells and they'd all be running off and doing their own thing. And how would you ever get an animal that was properly laid out and organized and coordinated, right? If each cell was making its own decisions. So, of course, there's this whole other process of signaling, and here's an idea of how it works. So you've got a bunch of cells here, and you'd like, in some embryo, and you'd like to start making them different from each other. Well, how do you do that? Well, often in an egg, in an early egg, there is a cell that forms that's already different because it's near one end of the egg or something like that. And we're going to pretend that cell is this one, and it's turned brown, and what that really means is it is going to turn on some genes that the other cells haven't turned on. And one of those genes codes for a protein, a little yellow protein that gets released, and it travels across this line of cells, and more of it sticks to the cells that are nearby, and less of it reaches the cells that are far away, because it's too hard to get there, okay? So these cells then now have different amounts of this stuff stuck to them. And when it sticks to them, it triggers changes in the cells. So the cells out here at the right, they don't change because they never saw the signal. They're still blue cells, but these other ones form all these other kinds of cells, okay? And out of these, taking the hammer to the car to find out which wire in the back is needed for the horn to sound or for the electricity to reach the headlights and so on. We're looking at signaling things. You damage here, you see an effect over there, you infer a signal. And here, a whole bunch of signals were found. Here's one of them. This is what it actually looks like, sort of when you look at the atomic level. Okay, it's a beautiful little protein. And it travels through tissue and lands over here on some cell that it reaches and binds to some receptors on the surface of that cell, and that cell changes its behavior as a result of that signal landing there, in the way I was just telling you. What happens then is that you've got these signals bound to the outside of the cell, and here's the chromosome, big tangle of DNA inside, and that signal is propagated into the inside of the cell, and a bunch of genes are turned on, and then those genes make their own kinds of proteins. And now here are a whole bunch of proteins that have been made, and now that cell has been changed into a cell with different properties and character. But remember the beginning of this, that because the signal travels in a certain path and reaches some cells and not others, you have a whole system of different cell types laid out. Some of the mutants that were discovered, they were involved in signaling, had all sorts of funny phenotypes, meaning funny appearances. One of them was very bristly, and it was called the hedgehog mutant. And it turns out that hedgehog signaling, which was discovered in flies, works in us. Same old story, ancient conserved thing in evolution. And for example, in the formation of our spinal cord, so that's a pretty important part of our nervous system, you need different neurons to form at different points around this circle of spinal cord. And this hedgehog signal comes out of the cells down here and signals up to each side and causes cells in particular positions around the spinal cord to take on certain fates, to form certain types of neurons. Motor neurons versus sensory neurons and so on, okay? And when you don't have, it also affects the forebrain, our pride and joy. And when you don't have this thing working right, you get cyclopic embryos that don't have the split of the brain into two hemispheres and have only a single eye. And in the early days of Carnegie studies of human development in 1917, these kinds of embryos were described. There are quite a few causes, but hedgehog damage to this hedgehog signaling system is one of those causes. Okay, so this is a signal that lands on a cell and turns on genes, and now we have modern genomics, which is the study of how batteries of genes get turned on and turned off. And so here's genomics. This is in the Falkland Islands. I couldn't believe that we were staying in this bed and breakfast, and I looked out the window. I said, this is too good to be true. But what genomics really means is looking at the organization and structures of genomes, and you can take some cells and dump some hedgehog signaling protein on them and say what genes come on in response to that. And what you find out is there are a whole bunch of different genes that all do all sorts of different things, but a couple of hundred of them are involved in forming various kinds of neurons. So you start to learn how one signal controls a whole set of responses. And I'm going to sort of summarize this about the signals by saying hundreds of genes can be controlled by a single kind of signaling protein. And since there are many other signaling proteins emitted at different times and places, you basically get cells responding to a sort of barrage of information coming at them. And that's what organizes in 3 dimensions and in time the differentiation of cells and embryos. Excessive hedgehog signaling can turn cells into cancer cells, which is what I'm going to finish with in a moment. It acts during, the same signal is used during the development and regeneration of all of our tissues and organs. It turns out these signals are like a toolkit that's brought out again and again during development. And there are other signals, as I just said, acting in parallel. So damage to signaling systems like this one that normally control growth, among other things, if the signal normally says grow, selectively, right time, right place, and now the signal gets out of control due to some mutation of genes, it's going to still say grow, but it's going to grow when there shouldn't be growth. And in fact, damage to this hedgehog pathway, as we showed in my lab in 1996, is the cause of the most common human cancer, which is basal cell carcinoma of the skin. It's also the cause of a brain tumor called medulloblastoma, which is the commonest childhood malignant brain tumor, terrible tumor. And what's happened now since is because we know a whole bunch of the switches and components of the hedgehog signaling process, how the signal is sent, how it's received, how it's interpreted. Companies are making drugs that target different parts of that signaling system to try to make anti-cancer drugs. And the first one of those was approved by the FDA about 2 years ago, happily on my birthday, which I thought was awfully nice. And it's interesting in terms of thinking about basic science to trace the course of events, because the genes, the relevant genes, were discovered in flies and reported in 1980. It took 16 years before the connection to cancer, first of all, the connection to mammals and us, and then the connection to cancer. That took 16 years more of research by many laboratories working hard. It took another 16 years to find a way to devise a drug that would affect this pathway. So it was a 32-year process to go from the basic science discovery in flies through these intermediate steps to the beginnings of some new therapies. Okay, now in one sense, everyone who has these diseases like this is very impatient, and rightly so. You'd like to cure it yesterday. But in the larger scale of things, 32 years is not all that long. And now, of course, that can be acceleration. And it's useful to accelerate because hedgehog signaling is not only involved in those cancers, but in some others to which it contributes. They're shown here: breast cancer, esophagus, liver, prostate, small cell lung carcinoma, pancreatic cancer. All of those can be exacerbated. The primary cause is not damage to hedgehog, but hedgehog kicks in and makes things worse. Okay? So again, examples then of how basic untargeted science, how does segmentation work in a fly? That was the starting question, and that led by a direct and logical process, not as fast as we like, but it got there, 32 years, to discovering a way to treat the most common human cancer. Okay? So when people get impatient about basic science, it's sort of understandable, but it is a process. It can take a long time, but it can lead in all sorts of fantastic directions eventually. Summary, and we're almost done. Life changes sometimes quickly under genetic control. Changes in the control of development underlie changes in animals and plants. The unity of life is reflected in genes whose roles in controlling development have lasted at least 500 million years. I've been telling you about these evolutionarily conserved genes. And understanding how the genes control form and function leads to new ideas for medical therapies and to new ideas about life and human origins. Now, the very last thing I want to show you, or actually allow myself to read to you, this is recording myself, but that way you don't have to look at boring text, you can look at the ocean. John Steinbeck wrote a beautiful passage, which I put in the current yearbook of the Carnegie Science Institution, in the book Log of the Sea of Cortez. This was a marine biology expedition he took with his friend Ed Ricketts, a very famous marine biologist whose beautiful book, Between Pacific Tides, is still in print to this day. They went on a boat called the Sea of Cortez and went there and collected a lot of specimens and got drunk a lot. And the book is a sort of diary of the trip with lots of interesting commentary, but it has this one passage which I think captures what the Carnegie Institution is about, which is this span that John alluded to from astronomy and cosmology on the one hand to ecology, the other with geophysics and geochemistry and plant and animal molecular and cellular biology in between. And that's what you can hear, I think, right now.
Our own interest lay in relationships of animal to animal. It seems apparent that species are only commas in a sentence, that each species is at once the point and the base of a pyramid, that all life is relational to the point where an Einsteinian relativity seems to emerge. And then, not only the meaning but the feeling about species grows misty. One merges into another. Groups melt into ecological groups until the time when what we know as life meets and enters what we think of as non-life. Barnacle and rock, rock and earth, earth and tree, tree and rain and air. Then one can come back to the microscope and the tide pool and the aquarium. But the little animals are found to be changed, no longer set apart and alone. And it is a strange thing that most of the feeling we call religious, most of the mystical outcrying, which is one of the most prized and used and desired reactions of our species, is really the understanding and attempt to say that man is related to the whole thing, related inextricably to all reality, known and unknowable. The profound feeling of it made a Jesus, a St. Augustine, a St. Francis, a Roger Bacon, a Charles Darwin, and an Einstein. Each of them, in his own tempo and with his own voice, discovered and reaffirmed with astonishment the knowledge that all things are one thing and that one thing is all things. Plankton, a shimmering phosphorescence on the sea, and the spinning planets and an expanding universe, all bound together by the elastic string of time. It is advisable to look from the tide pool to the stars and then back to the tide pool again.
Thank you.
Wonderful. We have time for a few questions.
And if we could have the lights on out there, yeah, thank you. Great. Okay, in the blue shirt.
Oh. I thought you made the analogy to a car, but there's a big difference. You see, with a car, you know exactly what you can do. You get out your hammer or your jackhammer, and you go bang, take out the engine, and you see what happens when you try to drive the car. It might blow up, might do that, and so it's pretty obvious what happens. You can't take out some DNA hammer and go bang to a gene. It isn't like that. Can you actually pinpoint like you can with a hammer and a car? Or do you have to just randomly throw x-rays and cross your fingers and hope really badly that it hits?
Okay, great. So several good questions there. Let me get behind the speaker. So can you, first of all, can you target a specific gene and hit that one? And the answer is when the experiments I was describing were first done, you couldn't. You did it randomly, which is the other thing you suggested. And then you looked for any damage that would, say, prevent muscle growth or something like that. And that allowed you to identify, and then you could find out where the damage had happened. Nowadays, you can actually engineer rather easily damage to a particular gene that you think might be the important one, and so you can test an idea like that in a very targeted way. The other part of your question was, you sort of know something about a car, but I was sort of postulating that suppose you had never seen a car and you were approaching it and curious about it, much as we've never seen the genes that are needed to build an embryo. So it was a somewhat hypothetical thing, and I'd really like you not to go outside to any of those cars out there and do what I suggested. Yeah, okay. Yeah.
All right, thank you for a great presentation.
You're welcome. Thank you.
So it must be true that there are some ancient conserved genes that have been identified that date all the way back to before we animals split off from plants. Could you talk about that a little?
Yeah, so the question is, are there genes that are older than the start of, the animal-plant difference or the animal-plant-fungus difference? And indeed, the genomic studies are now completely revamping our ideas about early life. We're with the plants on one branch, and then there are all these things, that vast complexity where things are as different, much more different from each other than we are from plants. But all of the metabolic apparatus, the enzymes for processing sugars and DNA and all these catalysts and so on, those are all there in bacteria, and they're pretty much the same as the enzymes in us. So those are billions of years old, and they're of course in plants and in us. So yes, there are things. And then just to balance it a little bit, there are things that are more recently evolved that we don't share with other animals, let alone with plants. We have a very complex immune system that's adaptive. So during our lives, that's how vaccines work. Once we see something, we produce a lot of antibodies that can recognize that thing that we didn't like on the first pass and never want to see again, never want to deal with again. So we actually adapt our immune system. That whole immune system involves a whole bunch of genes that are not found in many other animals other than on our branch of the tree. Yeah.
I've noticed in my Science for Dummies magazines in the last, they talked a lot about epigenetics. Now, it's a factor on top of external.
It is. So epigenetics is the question. What's epigenetics? So genetics is the code in the DNA. You change those A's, C's, T's, and G's into something else, and you can inherit the something else. That's straight genetics. The very first slide I showed you about chromosomes, though I didn't actually point at it, it said chromatin, and it said there are proteins packed onto the DNA. It's like a whole complex packaging. And part of that packaging is simply to allow that tangle of DNA to compact in an organized way. So it's literally packaging. But some of those proteins are actually sitting there, keeping genes in an on or off state. And sometimes when you copy the DNA, the position of one of those proteins turning, say, this gene off is preserved when you make two copies of the DNA. So now you've got an inheritance that has nothing to do with any change in the DNA, but is the fact that the protein is, the position of the protein at that location is passed on. And so there are quite a few genes that are regulated, in fact, by these proteins that are glommed onto them and whose positions there are maintained. There can also be actually modifications of the DNA, chemical modifications of the DNA that are heritable. So there's a whole other layer on top of the inheritance of the primary DNA sequence that can be passed through at least multiple cell generations and sometimes many. Yes.
Are there alternate forms of life that you can imagine? I mean, everything on Earth seems to be fixed on this double helix and blah, 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 blah. But is that necessarily unique?
Okay, so could there be other machinery, essentially, for life other than the machinery we know well? So there are a lot of different interesting sides to that. So one is why isn't life still evolving? Why isn't life starting now? Again, a new kind, a different kind. Because why not? I mean, there's plenty of nutrients out there, and why not have whatever happened to start it the first time, which is really quite unknown. And evolution is often confused. Evolution is change in life, not the origin of life. So the origin of life problem is very tough. It's a problem in chemistry and so on. It's very speculative still, though there are some experiments. Anyway, one of the problems is there's so much life now that if any new life started up, it would be instantly in competition with very advanced forms of life that might simply eat it or outcompete it or whatever. So that's a problem in seeing it happening. But is there any clue, for example, to think about your question, for a different system? And I don't think we've seen anything totally different, but there are little elements of differences. There are certain proteins that pass on inheritance because the protein can flip from one shape to another. A protein that is flipped to a certain shape, when it comes into contact with another copy of that same protein, will cause the next one to flip into that shape, and that's passed on through many molecules. That's a different kind of inheritance, and for those of you who know about prion diseases, that's what those are. They can affect brain function, for example. And then there are many viruses that use RNA as the genetic code, not DNA. There's no DNA part of it at all. It's only RNA. So there are ideas of either the origin of life or an alternative origin of life where the primary molecule is not the DNA double helix, but the RNA molecule, which is a chemical variation of DNA. So not totally different, but somewhat different. And that's about all we know right now.
I mean, it's the same system.
Well, in some ways it's similar, but in other ways it's different. But I agree, that's not something completely different. You'd love to find something completely different, and no one has. But of course, that doesn't mean it doesn't exist, especially elsewhere.
Let's do one more question, and then.
Are there any multicellular organisms that don't have Hox genes?
Are there multicellular organisms that don't have Hox genes? Yes, there are a lot. So generally, the organisms that have Hox genes have some very clear organization of different structures along the head to tail axis. But the organisms that don't have genes that sort of give that information of patterning, they still make proteins that are related to the Hox proteins. They just do different stuff. So you can actually see, even in bacteria, you can see proteins. You know how I showed you how the protein docks against the DNA perfectly? There are proteins that have that same shape that are in bacteria. And they turn genes on and off. They just don't do it to put legs and wings and eyes in places.
So it'd be possible to have Hox genes in prokaryotes?
Well, I don't think we'd call them Hox genes because Hox genes really refers to something in positioning appendages and vertebrae and so on. So what we would call them is homeobox proteins or homeodomain proteins because they have this little structure to them that binds DNA, and that unifies all of the proteins in that family. Proteins can generally be grouped into structural groups where they have similar shapes that do similar things at the molecular level. But Hox then would be specifically referring to ones that control head-to-tail patterning. It's a great question.
I know there's other questions. If you want to, please come up. I'm sure Matt will ask them. Let's thank Matt again then.
All right, thanks. Thank you.
Realize that. I'm sure.